That's brilliant. Okay.
Yes.
Let's get my PowerPoint going. Yes, fantastic.
Thank you.
I'm trying to get these to share the screen. When I put that, it says host disabled participant screen sharing. Hello? Hello?
Sorry, doctor, I had not made your.
Huh?
Yes.
Okay, fine. Okay, let's see. Okay, so let's start. First of all, good evening, and thank you very much for indulging me. That's really brilliant, and I highly appreciate it. Nonetheless, what we have done is, and what has been fortunate, is to be able to literally go through the internet and look for PowerPoints that are appropriate, that have been done in detail for purposes of reading. Most of the stuff you get is there, but it has a logical sequence that one has to select after many, many years in teaching. Suffices to say, when you're in this fourth year, all your reading is geared to be able to make some clinical decisions based on some paraclinical science, of which immunology is one of them. So nonetheless, we're going to go through these things, and I will highlight things that we're going to be talking about and things that become apparent to you in your practice. So first and foremost, this is standard validity. So what we have to say when you say hypersensitivity is they are actually exaggerated responses to a otherwise normal or harmless antigen. And since they're grouped into four, in terms of urgency 1, 2, 3, and 4, majority of the times we look at type 4 and say, well, is it really for it to, for completeness sake? Nonetheless, you'll be seeing that more than often in your clinical practice. Suffices to say is that the one thing you want to do is avoid hypersensitive reactions in your practice. So these are the two areas that are very, very important for us to appreciate. Is type 1 where we have an allergy caused by environmental antigens, whereas autoimmune diseases where we have immune responses against actually extremes of a whole panorama of disease processes. Suffices to say, our actual goal is to avoid allergy and autoimmune diseases at all costs. Okay. Since we're talking about autoimmune diseases, which we'll talk about MHC. There's always an immunological or familial be manifest, but that's only in some, not entirely true. It only means that there's a higher association if you have allergy, if you have similar HLA or MHC, and for that matter, autoimmune diseases. Ultimately, what's really scary, or what we have to prevent or to actually combat, is the fact that these particular hypersensitivities have high morbidity and high mortality. And ours is to minimize any event as opposed to treat. Can you leave that alone? We don't use it anymore. That's okay. This is just looking at a diagrammatic representation where we're looking at comparing and contrasting all four in a tabulated diagrammatic form. Good for quick revision, but essentially not really important because we're going to highlight type 4. So principally it is caused by inflammation resulting from cytokines produced by CD4/8, CD4 plus T cells, and cytotoxicity by CD8 plus T cells. As you can see, both together constitute a T cell mediated hypersensitivity. Now, in contrast to types 1 and type 2, in type 1 we had a Th2, helps lymphocyte involvement. We had the B cells, we had effectively at the end mast cells and basophils. But here we have literally a cell-mediated immune response, as opposed to humoral in type 1 and type 2. So again, CD4 plus T cells mediate hypersensitivity, major role in autoimmunity, which will be discussed later. I hope they will do that. And CD8 plus T cell mediated hypersensitivity, predominantly against viral infection or endogenous antigens, tumor cells, and graft rejection. The brilliant thing about immunology, and it's always science based on limited foundations. It means that every immune cell has a particular function. This function is the same irrespective of whatever the disease process is. So if you talk about phagocytosis, you can phagocytose a tumor, a virus, a bacteria. The role remains the same, only the production of the name of the production or the logical production is different. Suffices to propagate it. Now, in CD4 plus T cell mediated hypersensitivity, cytokines produced by the T cells induce inflammation that may be chronic and destructive. This will be classically seen later as we go through the various slides. And the prototype of cell-mediated inflammation is delayed type hypersensitivity. Why the concept of delayed is because it can take anything between a couple of hours to days. Normally, in certain cases where we talk, it will be critically 24 to 72 hours. Suffices to say, it is delayed with respect to something like type 1 or type 2. In type 3, there's a massive delay of months to years. Now, what is crucial to appreciate, we have type 1, TH, and TH17. These are crucial, important T helper cell groups, especially the TH17, which is now gaining prominence. Okay, so inflammatory reactions stimulated by CD4 plus T cells can be divided into stages: activation of the CD4 plus T cell, followed by responses of differentiated effector T cells that will bring about the immune response. So let's look at this one. Activation of CD4 plus T cells. Classically, they recognize peptides displayed by an antigen. What's crucial is that the interleukin-2 recruits, enhances the recruitment and differentiation and proliferation of other T lymphocytes. It is crucial to establish and to recognize that in the initial contact between an APC and a CD4 plus T cell is that the CD4 plus T cell is naive. It has never been exposed to any kind of immune interaction. Once it is activated, it now goes and matures or starts its function along a particular path. It cannot be swayed. Now look at the impact of all of this. For an antigen-presenting cell to go into a lymph node, it literally has to go through the capsular region, the medulla, literally looking for its respective T4 cell. It can be a problem because there are almost about 100 million to a billion types of T cell clones. To prevent that, there are certain microenvironment features that prevent a T cell from exiting if it does not find a suitable T cell to engage. So your proliferation of CD4 plus T cells and activation of APCs, in this case dendritic cells, productions of cytokines, interleukin-12, 6, and 23. Nowadays, IL-22 and 23 are becoming very prominent, and these are the things that will be heralding a lot of science in your fifth year. So what do we get? If it's interleukin-12, differentiation into TH1. If you elaborate interleukin-6, you go to TH17 cells. Mark, it is important to realize that interleukin-6 is a very prominent pro-inflammatory cytokine of the innate adaptive, so the innate system, and is very, very prominent for some reason in ENT pathology. Of gamma interferon, further TH1 development, or more importantly, is this section. Th1 secretes gamma interferon that activates macrophages. This is the hallmark, as is this. And why is it so important? Is because there's sometimes there might be an antigen or a biochemical product, maybe an inert product like a piece of wood, very tiny piece of silica, coal, etc., that is difficult to digest by conventional phagocytosis. So by going through this pathway, we actually facilitate the augmentation of the phagocytic activity of a normalized normal macrophage. And that is the hallmark of the pathology. So if we go to the extreme left, we see increased ability to phagocytose and kill, and that is very crucial. All right. So activation, what makes this macrophage different from another one? Expression of more MSC class 2, facilitating further antigen presentation. Finally, secreting of our all too well-known interleukin-1 and alpha TNF, which are very, very aggressive pro-inflammatory cytokines of the so-called innate system. And finally, we have the interleukin-12. At this level, let us look at that diagram and see how we can synthesize that into a working formula that enables us to understand the very dynamics of this kind of progression. You want a macrophage to be aggressive. You want to enable it to be able to maintain its presence as opposed to its limited longevity sometimes. So this activated macrophage develops the biochemical and physiological apparatus to be able to sustain its existence in tissues for longer times, enable it to do its primary job, phagocytose and kill. It itself plays a role as an APC, furthering the cycle. You also want to secrete pro-inflammatory cytokines that attract other macrophages and other associated mononuclear cells. Finally, you want to amplify the Th1 response. So APC meets naive T cell. APC secretes interleukin-12, amplifies the Th1 response. Similarly, the Th1 itself has a sole function of producing gamma interferon that will activate macrophages to give you all those four remaining or four highlighted functions. And at the same time, also, the activated macrophage itself produces interleukin-12 that can act on both the APC and the Th1. This will be classically seen in TB. So you do the pathophysiology of TB, in essential what we're talking about is a type 4 delayed hypersensitivity reaction. So look at Th17, a newcomer to the theater, although not when I say newcomer, it's now very manifest in current 8th or 7th edition textbooks, but it was discovered over 8 years ago. Th17 secretes interleukin-17 and 22, chemokines and other cytokines. Critically, to do that, recruitment of neutrophils and monocytes. So when you're in autoimmune diseases, IL-17, IL-22 will play a critical role in pathogenesis, especially in things like, or disease entities like rheumatoid arthritis. So here we have a diagrammatic representation. All right. Between here and here, we have interleukin-12. All right. Between here and the macrophages, you have gamma interferon. Interleukin-12 here, from here to here and to there. But what's critical here is this tissue injury, which is now become of surrounding tissues, normal surrounding tissues, plus offending agents. This is classically manifest in the so-called tubercular reaction, all right? That was time-ordered in our times. We wanted to see whether or not you had an acute infection, a past infection, or a very exaggerated current infection. Suffices to say, we injected subcutaneously sensitized individual, someone who has received what? Beta, HCG, or what? What do you receive to immunize yourself against TB? BCG. So this is what it looks like when you actually take a histology of that tissue. Between 72 to 24 hours, 24 to 72 hours, you'll see a hardening of the skin tissue, very prominent hair follicle indentations, bit of tenderness. That was called induration. We used to measure the diameter of that, and using a standard 0 to 5, 5 to 10, greater than 15 millimeters diameter, we could decide upon whether it was acute, chronic, or a past infection. So here we are. This is This is immunohistochemistry. So you can see all these portions have got edema, widely separated places. Any fiber you can see as a very fineish, pinkish material. Perivascular cuffing, let me see, I get a good blood vessel. No, I'm not really sure about this one. But this could look like a blood vessel. Perivascular cuffing by mononuclear. We come to the crux of the matter, is now the actual pathology. So as you're taught in chronic inflammation, the mainstay or the characteristic cell that is associated with granuloma, in fact, for chronic inflammatory conditions, was the so-called appearance of a macrophage in the tissue. But we have to have an active, a very aggressive response. So we tend to get what is termed as granulomas. That is one of them, that's another, that's another. You can see the mononuclear cells are all over here. These seem to be walled off by thin tissue, fibrous tissue. All of this is a plasma, I think a giant cell here, if I'm not wrong. Yeah, I think one, yeah. And you get your hyperactive macrophages, you have collateral damage, destruction of cells, you have a fibrous kind of capsule loose, trying to localize that infection or the inflammatory response, and then a cuff of mononuclear cells. So examples, contact dermatitis, there are people that develop, they get a scratch with a bark or some kind of thing. They manifest not immediately like type 1, but they manifest after some time. Those are delayed type hypersensitivity reactions. It is critical to establish the time duration between type 1 and type 4 for purposes of classification. Rheumatoid arthritis, this is crucial because Th17 acts against collagen and citylated cell protein. Okay, so whatever it produces here is toxic to the cartilage. So with autoimmune diseases like rheumatoid arthritis, we want to be looking at a more exaggerated CMI, cell-mediated immune response, as opposed to an exaggerated humoral-mediated immune response, although we can produce autoantibodies against rheumatoid factor. Then other examples, multiple sclerosis, the now appearance of Th17. Now, cytotoxicity. That was discussed, I hope, in your preliminary foundation course in level 2. I talk about it that way because very soon I shall not be around with you guys entirely. I want to move on. So I'll be referring it to historically. Now, what's classical? CD8 plus cells. Here we have an antigen presenting cell with what? MHC and cupping an antigen. So that should be TH1 or TH2, it should be TH1. You activate the CD8 plus cells, you get destruction of tissue bearing the respective antigen self. So what is important to remember is this presentation is done directly. There isn't much of a cytokine elaboration because CD8 plus cells really have internal organelles and internal secretions of biochemical lytic compounds that are able to do its work. So it's actually not going to be significant if we have too much cytokine elaboration, although there might be some. Now, 3 different ways about which CD8 does cytotoxin mediated. Perforin granzymes are critical. All of these ones, especially this one and that one. I repeat, perforin and granzymes are really critical because when the 2 cells come into contact, let me go back to the top one, this CD8 plus cell will produce a molecule that resembles a cylinder, like the stuff you get when you have paper all around a cardboard cylindrical thing, or those which contain maps. It'll punch a hole between there and there. Just hold on, excuse me. Hello.
Hi.
Yeah, hello.
Where are you?
Home.
Home.
Yeah,
Can
bring us.
you pick me from the spa in another half an hour?
Fine, I'll do that. Let me go to my glass. So. That's perforin there. Along that particular so-called corridor or cylindrical 3-dimensional punch will be granzymes that goes from there into that cytoplasm. Or if it's a very aggressive response, these CD8 secrete granzymes directly all over the exterior and also at the same time through perforin. Perforin forms a kind of a corridor from one cytosol to another, along which granzymes are actually transported from the target cell, from the effector cell to the target cell. Okay, this is okay. This is the anything extra if you want to look at that. This is not, it is not actually examinable, but you can look at it if you want to. This is for pure interest. Now, here's what the most another point is. We talked about perforin, granzymes. Now we talk about caspases. Granzyme induces caspases, which is actually a kind of apoptotic enzyme. So there are 2 pathways we use to destroy. One, using granzymes, perforin. The third, you induce apoptosis by inducing caspases. And this is how it functions. Histone is very crucial in DNA. Yes, very crucial. So its target is histone. Perforin we've done. It's okay. We mentioned granulysin against gram-positive, gram-negative. It's more or less for infection, microbial causes. Let's see, these are the ones, don't worry. Good. Yes. This is the one we talked about. This is another way of inducing cell death by the fast ligand complement. Death receptors are crucial in immunology in 2 areas. In the pathogenesis of autoimmunity, it's one of the 3 notable pathways, or it can be used in direct type 4 reactions. Going back to autoimmunity, if a self-APC presents MHC and a self-antigen to a naive T cell clone that will react either to a high degree, severe or a moderate reaction, it will inhibit that particular T cell naive clone by actually having that cell express on its cell membrane FAS, CD95, and FASL, fast ligand, or CD95 ligand. Binding of those 2 induces apoptosis. So activation of CTL, CD8, protein, FSL. You engage the two. This one associate, this is part of the alpha TNF group that actually facilitates this. All right. Finally, you get production of these ones. These are not really important in terms of exams. It's just to give you a complete idea of how it works. Your job is to find out what is FADD. All right. In the meantime, let me just look and check what's happening. Good. Finally, we come to cytokines. This is very elaborate. This is not the scope of your training. This is for an immunologist or a basic scientist. Cytokine mediated, again, what do we have? Gamma interferon following viral infections and exposure to some contact. So how is it produced? The TCR or secret lines of antigen dependent or a minor pathway where you elaborate type 1 interferon and interleukin 12. It also induces gamma interferon. But STAT4 is a signaling pathway, antigen independent. So it makes CTL able. This interacts major pathway. Again, we're now looking very actively at gamma interferon as a particular immunotherapeutic agent that may be used in a variety of anti-tumors or anti-rejection systems, or even actually antimicrobials. This is type 1 or this is no. Now, this is self-explanatory, except here we have TH1 and TH17 cells. Then we have CD8 plus pathways. And this one comes to summarize viral infections, tumor, and graft rejection. With that in mind, so what kind of examination questions can we anticipate? Question number 1 can be a long, short answer question. First, define what is meant by type 4 hypersensitive reaction. 2, describe the immunopathogenesis of a type 4 hypersensitive reaction using a suitable example. Demonstrate the pathophysiology of a disease entity that you're well aware of, like TB. MCQs that are crucial. 1, the most important cell in a type 4 reaction, especially when you have Th cells involved, is an activated macrophage. You might be asked, what are the cytokines that are crucial in that reaction? You have gamma interferon, you have interleukin 12, etc. You may be asked the features of an activated macrophage are the following, except. You also may be asked, when do you get, what other conditions can give rise to a granuloma? All right. This you must actually investigate, because when you're going to be out there, there'll be times when an animal or a person, especially a person gets injured, they have a shard of wood in them or a splinter, they have a piece of bone for that matter, maybe a bit of a bullet or something, gets lodged in, we tend to form a granuloma, especially with respect to deep-seated mycoses of fungal infections. So you want to be looking at an example that you'll see if you're working in Northern Frontier District. This is the old colonial way of saying it, but in the northern areas of Kenya, especially with pastoralists, you'll have infections with something known as Madurella mycetoma. Madurella mycetoma that will give rise to a granuloma. These tend to be fungating, superimposed bacterial ulcers that never seem to heal. So if you're in such regions where you have pastoralists or you have a lot of acacia trees and animal dung, fungi will be actually quite prominent. Always asked to have a bit of the aspirate examined under a microscope without staining, then stain. And if you want to know that it's actually a fungal infection, you put your fluid, aspirated fluid, between two slides and you'll hear a crashing sound like grittle or grittling kind of sound. That will indicate what they call fungal elements, and also stain for them too. So remember that one, Majeuranomacytoma is a very famous thing. It's a deep cell mycosis, something that you'll actually see. All right. Another example of granuloma is when you have schistosomiasis, Mansoni, especially in the liver, where you can tend to get areas like when you have the portal tracts, etc. It's an immune reaction to the egg as opposed to the actual parasitic adult male or female. So, ladies and gentlemen, thank you very much. I sincerely hope this was eye-opening, and I hope that you don't forget this one because it is readily forgotten when you don't see that many things. Thank you very much and have a nice night.